that I introduce Dr. Agenor Limon. He is professor at the Neurology Department and the Mitchell Center for Neurodegenerative Diseases at the University of Texas Medical Branch. And today he will share with us his work in accessing functional and electrophysiological information about human synapses in both schizophrenia and Alzheimer's disease. These disorders have synapses as their major targets of pathological events. So the information that he's sharing with us today is of utmost importance to the development of new therapeutic strategies. Dr. Limon, thank you very much for being with us today, and you have the stage now. I think you are muted.
Yes, I think. Thank you very, very much for the invitation. It's really, really my honor to be here and share some of the work that we have been doing in the laboratory in the Mitchell Center, where I belong. And really, really thank you for the invitation. So here I have my name and the email where you can contact me at any time. And just diving in already. Let me see here. Yeah. So the overview of the talk is going to be, first of all, talking about the problem of understanding synaptic dysfunction in human disorders, how we are addressing that problem by reactivating receptors from human brains, how we are using the electrophysiological readings of synaptic receptors to understand synaptic dysfunction in schizophrenia for excitation, and then I will use that information to determine how we can measure the excitation to inhibition balance across brain regions in Alzheimer's disease. So all this information will be building up on that, and these will be examples of the work that we are doing in the laboratory. So, as you know, during all our lives, we are susceptible to many types of brain alterations. So during the childhood, we have a high 2% of the population, the child population, is diagnosed with a autism spectrum disorder. 1% of the population in adolescence and early adulthood is diagnosed with schizophrenia. Then we can have bouts of medical depression. And when we are getting old, aging is the highest risk for developing Alzheimer's disease. So across all our lives, so we have different levels of susceptibility for the functioning of the brain. And we are not taking in consideration many others, for example, epilepsy or alterations that are going to produce behavioral changes and difficulties in daily life. What all of these have in common is that these behavioral alterations are produced by the electrical activity of the brain. So if we don't have changes in the electrical activity of the brain that is going to modify our behavior, then we don't have these problems. So despite all the different behavioral alterations, these are resulting of the electrical, resulting from alterations in the electrical activity of the brain. Another important part is that the electrical function of the brain is determined very, very strongly by the neuronal communication, or by the communication between different cells. The synapses that are represented in this cartoon is basically the way that the neurons communicate to each other by electrical and chemical signals. And it's in these synapses, the release of these neurotransmitters that activate different types of receptors, among them GABA receptors, AMPA receptors, that determine the electrical activity of the brain. So it is not a surprise that alterations in synaptic number are going to be associated with alterations in brain function. And this slide, I like it very much because it represents many of the changes that are associated with the structural changes in the neurons. Dendritic spine number in the cortex are representative of excitatory synapses. And as you can see in the black line, we have different numbers of excitatory spine across our life. Before birth, we have an increase of this number of synapses, and then we have a plateau. They start to reduce during adolescence, middle age, and then normally we continue to have a reduction of these dendritic spines across our life. In Alzheimer's disease, we have a normal number of synapses, And then in middle adulthood, we start to lose these synapses. And in some cases, it's going to produce mild cognitive impairment. And then during the last stages of the severity of Alzheimer's disease, we have a large reduction of this number of synapses that leads to dementia. In schizophrenia, the change here is a little bit earlier, and therefore the symptoms that we're going to observe in schizophrenia and Alzheimer's disease is going to depend on the different brain regions that are affected and the timing where this synaptic loss is observed. In autism, we have the opposite situation. We have an increasing number of synapses that is associated with loss of pruning of these synapses. That is going to have an increased number of communication, more excitation, and more signal-to-noise ratio in the processing of sensory information and brain activity. And this number of synapses remain large across all our life, producing the symptoms or classical symptoms of the autism spectrum behavior. So now, because all of these disorders are not associated with a single gene, these are all multifactorial. So all of these are going to have multiple genes involved. Environmental factors are going to be also involved in this situation. Health, lifestyle changes sometimes affect. So how are we going to study the synapses alterations that are happening in these brains that determine the electrical activity of the brain, so we can determine what are the best therapeutic approaches? What are the best therapeutic approaches for all these treatments? So if you are in the field and you are trying to study, for example, Alzheimer's disease, we have also another problem. We have, in Alzheimer's disease, just recently, we have 210 animal models of familial Alzheimer's disease, meaning that we have mutations that are associated, or mutations that generate Alzheimer's disease. We have 20 animal models of Parkinson's disease. Now, these 210 models all are presenting different characteristics. And familial Alzheimer's disease accounts for about 5% of all Alzheimer's disease cases, because the majority of Alzheimer's disease cases are of the sporadic nature, meaning that there is not a single gene that is going to be producing the disease. So when you have to choose this animal model, what is going to be the best choice? So it has to be very well thought, because you're going to stay with that model for a while. So the question is difficult and very relevant. So because that is one of the problems in studying synaptic dysfunction, my laboratory is studying directly the synapses from the human brains. So then we can determine what are the parameters and endpoints of that we can measure in the synapses in humans, so we can choose better the animal models that mechanistically can give us insight in their function. I'm going to give examples now about the schizophrenia and Alzheimer's disease work. So here I'm just showing examples of the human brain in schizophrenia in healthy twins and in Alzheimer's disease. So healthy twins, identical twins, they have exactly the same information, but one has no, in this case, one has no disorder and the other has schizophrenia. And you can see that the large changes in the gray matter, as you can see here, enlargement of the ventricles in this case, and all this enlargement is going to be due to synaptic and neuronal loss. Below you are having examples of control of brain. In this case, neurons were stained with silver staining. So you can see the spines, that remember the spines are excitatory, and then you can see all these spines that are in these neurons. And in schizophrenia cases, there is a dramatic reduction of the number of spines, suggesting strongly that there is a reduction in the number of excitatory inputs in the neurons of schizophrenia. In Alzheimer's disease, you here have, again, at the right side, an Alzheimer's disease in very advanced stage, where you have cortical alterations, reduction, enlargement of the ventricles, atrophy of the hippocampus, and we also observe synaptic loss. So this is a control brain, and this is an Alzheimer's disease brain, where you can see the number of synapses being dramatically reduced. So we can think that just the synaptic loss is going to be enough to produce behavioral alterations. However, we have information that says that we are always losing synapses. We are always losing neurons. So this is just a plot where you have the age, the different colors indicates the different individuals, males and females, and then the ones that are associated with the line are the same individual taken at different times by MRI studies. And this is the thickness of the cortex. As you can see, this is in millimeters. So normally from childhood to aging, we are losing the thickness of the gray matter, indicating that we are losing neurons and we are losing synapses. However, in all these cases, cognition was preserved. There is no problem with cognition. So we can actually lose 1/3 of the thickness of the brain and still will have normal cognition. Then we also have in the brain synaptic plasticity and brain reorganization. That is how we are learning all the time. So in this example, I am showing data from 4 individuals that they were in late childhood and adolescence, but because tumors and epilepsy, they have to remove a whole hemisphere. And what happened is, of course, after the surgery, they need a lot of rehabilitation and therapy. But after a while, they were able to have an almost normal life. So that means that you can have acute damage in the brain, and if there is no chronic alteration, plasticity can overcome the destruction, and then you may have a relatively normal life. That is not the case of what is happening in Alzheimer's disease. Then we also have another problem. So the presence of severe neuropathological change in Alzheimer's disease is not 100% predictive of clinical dementia. And this is evidenced by resilient individuals. So what do I mean by AD neuropathological change? Is that the hallmarks of Alzheimer's disease are the presence of A-beta amyloid plaques and neurofibrillary tangles. Those are the toxic proteins that lead to Alzheimer's disease. But also, we have reductions in the number of synapses. So neurodegeneration is actually the strongest predictor of cognitive impairment. However, you can have individuals where the pathology and the amount of plaques and neurofibrillary tangles is very, very high, very severe. So pathologically, those individuals should have Alzheimer's disease. However, they don't have any cognitive symptoms. Those are individuals called resilient. And those individuals indicate that even in the presence of severe pathology, the brain may be compensating the changes and have normal cognition. Then the opposite is also true. There are people that are clinically diagnosed with Alzheimer's disease because they have problems in memory. But when you go to the pathology in autopsy and see whether these individuals have the hallmarks of the Alzheimer's pathology, they do not have it. So indicating that there is a mismatch between memory impairment and the pathology of Alzheimer's disease. And you may have alterations that resemble Alzheimer's disease because of other problems, neurodegeneration, some things associated with epilepsy, trauma, and so on. So because of all these alterations, then the question is, how are we going to study functional problems that are fundamental for neuronal activity and are the target of disease-specific remodeling processes? So we do microtransplantation of synaptic receptors. So microtransplantation of membranes was done originally by the group of Riccardo Miledi. Riccardo Miledi is well known as by the calcium theory of synaptic release, was working with Nobel Prize Katz, and a lot of work what Katz and Miledi was related to neurotransmission. We also started to do the heterologous expression of synaptic receptors to study electrical activity in xenophosphocytes. That's why you see in this photograph, he has a frog because all the work that he was doing, and it was a gift in one of his birthdays at the University of La Sapienza in Italy. So I did my postdoctoral studies with him, and I learned the technique, and we applied them to synapses. So the technique is basically this one. So you have a piece of brain that it can be fresh or frozen. Most of our work comes from frozen brain tissue. So this frozen brain tissue is homogenized to isolate the synaptosomes, and then we break them up to collect protoliposomes. These protoliposomes contain the synapses, synaptic proteins, and that they are embedded in the human lipids, and they are also associated to accessory proteins and are very relevant of the disease because these proteins were actually working in the human brain before death. Now, these protoliposomes are injected into frog oocytes, and once they are injected, similar to the one the vaccines are put in the new vaccines of mRNA are put in the cells, protoliposomes containing the proteins fuse with the membrane of the oocyte, and now these membranes can be activated. So the cell, the living cell of the oocyte, provides a living environment for the proteins that were transplanted. And then we can just activate them with agonists like GABA, glutamate, AMPA, and then all the recordings that we are observing are because of the human receptors. This happens because the oocyte per se, they don't have GABA receptors, they don't have glutamate receptors, so there is no way we can activate anything endogenous. So all the responses come from the human receptors. This is just an example of how, this is an example of an oocyte that was cut transversally, and this is a cartoon that represents the pipette with how we inject the membranes, and the membranes will be labeled with fluorescence. This is rat membranes. We don't use human tissue to visualization or other experiments that are just for showing purposes because human tissue is very, very valuable. So in this case, we use a rat cortex. And you can see how the membranes are deposited very close to the surface, and then they start to fuse and then laterally diffuse across the membrane of the oocyte. So by this, we know the place we are injecting in the oocyte because it's 1 millimeter size, and then we can do a lot of electrophysiological experiments on them. In this other side, I am showing membranes from torpedo fish that are highly enriched in nicotinic receptors. And then we apply alpha-bunker toxin that is labeled, and it can bind from the receptors just from the outside. As you can see, this indicates, the label indicates that we have nicotinic receptors fused to the membrane in the correct orientation, and then the bunker toxin from the outside can activate. If alpha-bunker toxin can bind it from the outside, it means that also can be receptors can be activated by endogenous agonists or any pharmacological treatment that we provide them. This is a representative example of how we do the control of the quality control of the synaptosomes. So we enriching in our preparations in synaptosomes. This is PSC95 that anchors excitatory synapses at the synapse. These are examples of the synapse that we can observe for synaptic densities, and then we break them up and fuse them into the oocyte. To the right, we have the responses that classically we obtain in a single oocyte that we inject with membranes. So we have receptors that are activated by GABA. We have receptors that are activated by kainate, and we know that the receptors that are activated by kainate are mostly AMPA receptors because we have done the pharmacological determination, but we use kainate because kainate keeps the receptor in an open conformation, so we don't have to apply other drugs to remove the desensitization that is classically observed in AMPA receptors, and it allows us to study AMPA receptors with a single dose of a drug. Then we also have NMDA receptors that we co-activate with lysine, and calcium channels that we activate by voltage pulses. All of this can be observed in the same oocyte, and as you can have here, we can also do pharmacological antagonism. All these receptors, as you can see here, we treated with ketamine, that is a specific antagonist of NMDA receptors, as you can see in these experiments. Nor GABA nor AMPA receptors were susceptible to ketamine, but NMDA receptors are blocked by ketamine. Because all of these responses can be determined in a single oocyte, then we can obtain ratios of all of these receptors responses, and then we can measure kinetics like activation, desensitization, and so on. We do many oocytes per single subject, so we can have variability, we can determine the variability, and then we do these responses in many subjects so that we can understand the variability of individuals across our diagnostic groups. So how we started to do these experiments to understand schizophrenia. So as you remember in my previous slide, I said in schizophrenia there is alterations in synaptic. So this is classic work from Dr. Glantz and Lewis, where it's showing the reduction in the spines in the dorsolateral prefrontal cortex of schizophrenia individuals. So in order to understand better the alterations of synaptic function in these individuals, we studied a small cohort of 10 controls, 10 individuals. These are the genders, the postmortem interval, pH, and ring, that is the RNA integrity number. And then we obtain a part of the dorsolateral prefrontal cortex from frozen tissue. We isolate RNA, and then we do transcriptomics using RNA sequencing. From adjacent tissue of that area, we also obtain synaptosomal preparations, and we did microtransplantation of synaptic membranes and also proteomics, label-free proteomics, to determine all the proteins that were present in the preparations that we are recording. Doing a very long story short, this is what we have when we do RNA sequence and proteomics of the dorsolateral frontal cortex. We expect to find genes that are upregulated in schizophrenia, genes that are downregulated in schizophrenia. And also we expect to have proteins that are upregulated in schizophrenia and proteins that are downregulated in schizophrenia. So this is the classical way how most groups do research using transcriptomic and proteomics. And then all the genes that are upregulated are processed by gene ontology to determine which pathways are enriched for all of these ones. As you can see, the genes that are upregulated in schizophrenia are associated to stress, glucocorticoid receptor signaling pathway, intrinsic apoptotic signaling pathways, and so on. Different pathways associated with stress and apoptosis are upregulated in schizophrenia. That is expected. Importantly, you can see that the most significantly represented pathways are synaptic ones. The genes that are downregulated are synaptic signaling, synaptic release, transmission across chemical synapses. This indicates that the instructions to generate the proteins that are involved in synaptic transmission are down in schizophrenia. And that correlates perfectly with the loss of synaptic responses, with the loss of spines that we have in the dorsolateral frontal cortex. So this is actually very reassuring that we can see that at the transcriptomic level. And when we go to the proteins that are upregulated, we observe some pathways that are already, that have been represented in schizophrenia, and has to do again with immune alterations, defend to other effects, and again associated with stress. And we have some pathways that are downregulated, for example, neurotransmitter uptake. So from here, we can see that genes that are downregulated produce proteins that are downregulated. Some of them are going to be associated with synaptic activity. And the proteins that are upregulated are going to be associated with stress. Now, once we have this information, we transplanted the preparations, and this is work done by Tommaso Cepilio. Tommaso Cepilio, he did a master in the University of Trieste in Italy, in the International Master of Neuroscience, and then he went to Texas to do a stay with me of 1 year. So in that, about 8 months, but he stayed 1 year, and he recorded the activity of these control individuals, and also a larger study that he's going to be co-author. But for this study, you can see reduction in the activity of the AMPA receptors. So you have small receptors, and we also have changes on the kinetics, but that's a different story. So this amplitude on the responses actually is significantly reduced. Every point is a single individual of these 20 individuals cohort. So there is a reduction that is significant for the AMPA excitatory responses in schizophrenia. Then he also did those responses of these receptors in all of the individuals, and then we have here the differences. So the reductions of the amplitude is associated to changes in number, but also it changes on the affinity of the receptors to AMPA, and in this case to kainate. So in purple, we have the schizophrenia individuals that are a little bit more to the right, and then in control that we have a little bit more to the left. So the thick lines are the average, and the thin lines is the every single curve for every single individual. You can see here that one individual with schizophrenia had a very low sensitivity to AMPA and kainate, and that is because we found a mutation on accessory protein and alterations in this accessory protein, TARP8, on this individual. So this indicates that the functional responses that we are having in our responses actually are representative of what is happening in the human brain. Because we have the proteomics and we have also the transcriptomics, then we started to see, okay, what are the proteins that are correlated and are producing our responses that we have in the synapses. So as you can see here, we found that the proteins that are positively correlated with AMPA receptors in our preparations. So here we have 3 plots, and in the common of this plot is the amplitude of AMPA responses here in the y-axis. Then here we have different proteins like RPS19, NDUF5, and GRIA3. GRIA3 is one of the subunits that generate the AMPA receptors. So AMPA receptors are tetrameric proteins made by the combinations of GRIA1, 2, 3, and 4. But also now we have information that they must have accessory proteins like cornichons, stargazing, TARPs. So this is the correlation. Again, all the individuals, you have the control individuals and the schizophrenic people with schizophrenia, and then you can see the correlation here. So as we expected, the more proteins that generate the responses or that generate the receptors, the larger the amplitude of the responses. But also we observe correlations with other proteins. As you can see here by protein-protein interaction modules, where we observe what are the proteins that are correlated with our responses, but also are able to be correlated within themselves and make more complexes. So all these proteins that correlate with receptors are important for the ribosomal proteins because we have synthesis in the synapses of, and energy management. Proteins that, complexes that are involved in mitochondria, external membrane of the mitochondria, inner membrane of the mitochondria, and AMPA receptor subunits. For example, via subunit AMPA2, via subunit AMPA3, and TARP8. This is the name of the genes for those proteins. So this is reassuring because, again, so we see the correlations between the proteins that we are measuring in proteomics and the function. Then we also observe that there are negative proteins, so there are proteins that are negatively correlated with the amplitude of the responses. You have here these 3 proteins that are involved in traffic, folding, and signaling, and the larger is the expression of these subunits, the lower is the amplitude of the responses, indicating that they may have a negative deleterious effect on the amplitude of the responses when they are increased. With all this information then, we started to integrate in a multidimensional analysis. And if we didn't have functional data, this is what we usually will have. The genes that are then regulated in schizophrenia and the proteins that are then regulated in schizophrenia. In this circus plot, we have the, in orange, we have the length and the number of the proteins that are upregulated in schizophrenia or downregulated in schizophrenia. And in here, in orange, we have the same genes that overlap one or the other. And in blue, genes that, although are not the same, they participate in the same pathways. You can see the number of genes that are altered in schizophrenia, up or down, is larger than the number of proteins that we observe in the synapses. And the number of correlations that we have are in proteins, and for example, in genes that are up and proteins that are up, is larger than the number of genes that really overlap. When we now include the functional information, and in this case, I am only showing the proteins that are negatively correlated with response in schizophrenia, then we get this picture. Now we have here the number of proteins that are positively correlated with AMPA currents, and if you remember, those are synaptic proteins. And then we have proteins that are negatively correlated with the AMPA responses, and those are proteins that are involved in trafficking and folding. So with this information, you can see then that genes that are downregulated in schizophrenia produce proteins that are downregulated in schizophrenia here. So genes that are downregulated in schizophrenia, proteins that are downregulated in schizophrenia, and those are the ones that produce the responses here. So the overlap is now much larger. And then proteins that are, so genes that are producing proteins that are up in schizophrenia and also negatively correlated with AMPA responses. So these have deleterious effect. And now the pathways that we observe are mostly enriched. So we call these electrophysiologically anchored dataset analysis. And as you can see, it provides more info, 3 different layers of information. And using these 3 layers of information, we can now find what are the pathways that are most involved in schizophrenia and the direction. So here you can see a MAPK. MAPK is a pathway that has been reported to be involved in schizophrenia, but whether this was positive, compensatory, or deleterious is not known. So this is the levels of evidence that we have for this pathway, like RNA sequencing, proteomics, and functional data. And all these indications put together tells us that increase of MAPK actually is going to be deleterious for the functional activity of the receptors. So then if we know that information, we can start devising methods to control or modulate negatively MAPK signaling in schizophrenia. So for the purpose of this talk, the conclusion is basically that AMPA receptors for frozen brains can be reactivated and recorded by 2 electrode voltage clamp. That is a technique that we use to activate the receptors. Second, that the amplitude of synaptic receptors currents correlate with the synaptic elements at the proteomic level. And third, that the function of receptors provide global directionality to signaling pathways that are commonly found altered in brain disorders. With this information, then we move to another problem that is a little bit complex. And that is because in Alzheimer's disease, as you remember, there is a reduction in the spines. However, these spines will suggest that we have less excitatory inputs in the brain. But what we observe is that the brain presents a hyperexcitability. So there is a larger prevalence of epilepsy. There is measurements of hyperexcitability in MRI and in electroencephalography. So if we have reduced excitability, how can we have hyperexcitability? So some explanations have been associated with how the circuits are connected, but a clear evidence has not been obtained. What we need to remember also is that all the spines are excitatory, and we can see them in silver staining, but what we cannot see in silver staining is the inhibition. So all the neurons receive excitatory and inhibitory synapses. Inhibitory synapses I have anchor proteins like PH95 for the excitatory ones that anchors NMDA and AMPA receptors, and the inhibitory ones has anchors Gefferin that puts together in place GABA receptors. So there is a strong homeostatic control to keep the number of excitatory inputs and inhibitory inputs balanced in all the neurons. And this is very nice work by Barrals and Reyes, where we can say that the synaptic strength measured by the amplitude of the excitatory and synaptic inputs in a single neuron is correlated with the number of connections that that neuron receives. So the more connection a neuron receives, the lower is the amplitude of the responses, as you can see here. The lower is the number of connections, the higher is the amplitude or the synaptic strength of the connections, in order to keep a balance and keep the neurons in a set point that allows normal firing. There is actually a law that correlates the synaptic strength with the number of connections. So when in Alzheimer's disease, we are observing synaptic loss, we actually don't know whether the total inputs to the neuron is going to be a reduction excitation, because we need to measure also inhibition, and we need to measure how the remaining synapses is going to be weakened or strengthened by compensatory mechanisms. So the only way to do that is actually to measure directly the number and activity of the excitatory and inhibitory receptors. So this is Pietro Scaduto, that he did the PhD in my laboratory. He also came from Italy. He did a PhD in UTMB. And one of the first questions that we have to address is, when we are using human receptors, how protected the receptors are from postmortem degradation. And here we have, we did experiments in rat and mice. I am just showing here the experiments on humans. So we obtained autopsy brain tissue that we kept at different times to see whether we have the receptors are still active in these preparations, so we can record, and whether the activity of the excitatory and inhibitory remains constant. So we have 4 degrees, 15 hours in the fridge at 15 hours in the fridge, 27 hours, and 87 hours. We did 87 hours because we wanted to basically make sure that we have a point where everything was gone. But as you can see here, we were able to activate responses in all of the preparations. So there are changes of kinetics, as you can see here, and there is reduction of the amplitude, but it's not enough to not record. So actually we can record from all of them, and we can have responses measured by AMPA receptors and GABA. So we measure the amplitude. In the paper, you can see all the other analysis that we did. But for the purpose of this talk and the data that I'm going to show ahead, we measure the amplitude of the excitatory responses divided by the amplitude of the inhibitory responses. And as you can see, there were changes across time, but the changes were maintained across. So when we do the division or the ratio of these responses, there is no actually change in the excitatory to inhibitory ratio that we call electrophysiological EI ratio. So up to 87 hours. In order to study excitation to inhibition then in the Alzheimer's disease brain, we focus on the parietal cortex. There is data from MRI in the parietal cortex is some of the earliest stages or early areas where we have A beta accumulation. And also we have a associated excitatory to inhibitory changes observed by functional MRI in this area. So we took a frozen brain from the brain banks, and we took slices, and we did 2 types of analysis. We did fluorescence deconvolution tomography in collaboration with Dr. Julie Lautenberg and Gede Lynch. And the idea here is to label excitatory and inhibitory postsynaptic densities, and then count all the synapses automatically by software, where we just count the volume, so that we only count the immune labeling in volumes that are equivalent to synapses. And from an adjacent slide of tissue, we prepare synaptosomes, and we inject it into the oocytes to record their activity. So this is what we observed. So this is we did control individuals Alzheimer individuals, and we also include Down syndrome, because Down syndrome individuals have trisomy of the chromosome 21 that has the gene for the production of A beta. So because they have 3 chromosomes, they have triple the production of A beta, and they have a high risk of having Alzheimer's disease. Most of the people with Down syndrome around 50, 56, 60 years old develop Alzheimer's disease. So here you can see the labeling on type of in green of postsynaptic density for excitatory synapses, and in red for inhibitory synapses, GABA. By measuring only the volumes of the synapses, we can remove a large labeling of neurolytic, the dystrophy, lipofuscin, and other confounding factors. Here we have the histogram of distribution on the percentage of population, and here is the intensity of the labeling. So as you can see here, we have 5 individuals in each group and 6 actually in Down syndrome. So what we have here is that there is lower intensity of the synapses in Alzheimer's disease and controls when you compare to the control in gray. And for postsynaptic densities, we didn't have changes in the proportion. So actually, despite we are having loss on the reduction of the spines, because the brain shrinks, the number of postsynaptic densities are well maintained in Alzheimer's and Down syndrome. Then for the inhibitory ones, we are observing also shifts on the intensity, but here we are observing reductions on the percentage of the population for inhibitory ones. So then if we take the ratio of the amplitude of this divided by this, we observe that in Alzheimer's disease, there are a significant increase of this postsynaptic ratio of excitatory to inhibitory postsynaptic densities. In Down syndrome, we have a larger variability, and I will discuss this variability later. But only Alzheimer's disease compared to control show significantly differences in the layer 2 of the cortical layer of the parietal cortex in Alzheimer's disease. So from this data, we know that there are shared global synaptic disturbances in Alzheimer's disease and Down syndrome that include large synapse loss and changes of synaptic size, but the anatomical EI ratio in the parietal cortex is only increased significantly in Alzheimer's disease. By recording electrical activity of the receptors, then we have here at the same time GABA responses, kinase responses, and here I'm showing how kinase responses are identical to AMPA responses when we apply AMPA and cyclothiazide. So this confirms that our responses when we activate kinase are representing AMPA receptors. You can see that in Alzheimer's disease, we have a large reduction of GABA responses and AMPA receptors. So if you see the size here, this is 10 nanoamperes and this is 1 nanoampere. And here we have Down syndrome, that we have large increases. Actually, Down syndrome have the largest increase of responses compared to controls and Alzheimer's disease. But when we take the ratio is only increased in Alzheimer's disease and is not increasing Down syndrome. So this indicates that there are compensatory mechanisms of Down syndrome that increase the amplitude of the responses. But only in Alzheimer's disease, we have a higher participation of excitation. So regardless of the synaptic loss and reduction of the spines, reduction of responses, this reduction is not equal. So we have a larger and more dramatically reduction of AMPA responses compared to the inhibitory ones. We also observed in a cohort from the Allen Institute, we measured the DLG, that is the gene for excitation, for excitatory synapses, and Gefrin, that is the gene for inhibitory synapses. And we observed the same. So in Alzheimer's disease, we have an increase of transcriptional instructions for more postsynaptic densities. And the difference comes because of reduction of inhibitory ones, not of the excitations. And as you can see, this DLG and this Gefrin is actually very representative of postsynaptic densities when we do gene ontology. To understand why we have all these differences in abundance of excitatory synapses and inhibitory synapses, we went to measure in the Allen Institute the number of excitatory synapses and inhibitory synapses, sorry, the number of excitatory neurons and inhibitory neurons by measuring transporters for glutamate and for GABA in controls and Alzheimer's disease in the parietal cortex. As you can see, we observe the same. The cellular EI ratio, counting the number of cells excitatory to inhibitory, is increased in Alzheimer's disease due to reductions in inhibitory neurons. And when we do the ratio, actually the ratio, that cellular ratio and the postsynaptic ratio by transcription correlates very nicely. And the correlation is driven by Alzheimer's disease individuals. When we did this for this EI ratio, functional ratio for other regions, we observed changes in the parietal cortex that I just mentioned. We observed opposite changes in the frontal cortex and a large increase in temporal cortex. And in the hippocampus was very variable to make sense of those data. We also measure the temporal cortex excitation EI ratio in the temporal cortex in Alzheimer's disease, now measuring control, mild cognitive impairment, and Alzheimer's disease. And this was to observe if there was a gradual increase in the excitation to inhibition ratio in all these groups. What we observed then there is a gradual increase. And if the EI ratio is correlated with cognitive performance, so 30 are controls, and these are Alzheimer's disease individuals, and these are mild cognitive impairment. So the lowest we have the cognitive performance, the highest was the excitation, the electrophysiological EI ratio. And the higher the EI ratio, also the higher the A-beta protein, the higher the scores of A-beta plaques, the higher the score of the tau that produce the neurofibrillary tangles, and the higher the score of the spreading in the brain. By integrating this information with functional data, cognitive score, we observed two different groups in multidimensional analysis. So one group of proteins actually is the all good for synapses. So in this group, it was higher responses of GABA, higher responses of AMPA were actually correlated with higher cognitive performance and lower pathological alterations. And in the other group that we call the toxicity subgroup of proteins, or cluster of proteins, we have lower cognitive, lower amplitude of the responses, higher excitation and inhibition is correlated with low cognition and higher severity of Bragg stage and Serral stage that are markers of pathological change. This is just to finalize. This is another set of data where we are seeing now correlations of all our excitation inhibition values, cellular, transcriptional, functional, in this case it's transcriptional and cellular, and how it correlated with memory, executive function, language, and visual spatial data. The red is actually high positive, and the blue is negatively correlated. In this example that you can see here, you can see how memory, particularly memory, is correlated with differences in cellular ratio and transcriptional EI ratio in different brain areas. So this actually indicates that synaptic loss per se is not determining the cognitive alterations. So the conclusions are that in the parietal cortex and the temporal cortex, there is a strong loss of synaptic that are excitatory and inhibitory. Then the impairment of synapses is not equal, that leads to increased transcriptional, anatomical, and functional synaptic imbalance. And then pro-excitatory imbalance in ALD may explain hyperexcitability that we observe in the brain. And this hyperexcitability may interact with all the encoding of memory that we observe in Alzheimer's disease. So we have that. I'm just showing examples of schizophrenia and Alzheimer's disease, but we have many disorders that needs to be investigated. And I think other labs can start to do experiments or join a collaborative efforts. I'm just going to thank the funders. This is my lab right now. Some of the people like Pietro and Tommaso recording human receptors, and the funders like the National Institute of Health and my collaborators that were part of this work. And thank you very much. I'm going to have your questions.
Yes, thank you very much, Dr. Limon, for this great talk. Again, I would like to encourage our audience to write their questions in the Q&A. And we already have one first question from Catalina. She says, hello, your presentation was an amazing one and informative. My question is, what challenges and future directions do you foresee in the field of synaptic research in schizophrenia and Alzheimer's disease? And how might these insights impact the development of more effective treatments? Thank you.
Thank you. No, thank you for an excellent and great question. Yes, so some of the challenges that we have is access to brain. So in order to do this type of experiments, we have different things that have to be in place. First is obviously have the capacity of record big cells, like xenopus oocytes by voltage clamp. But the most important is having access to human brains that are frozen. So right now we have a strong collaborations with different brain banks in United States. We don't have collaborations in Europe yet, but in the United States we have collaborations with the University of Washington. We are also capturing our own brains by donated brains in Texas. In University of Washington, we are working with different groups collecting those brains. So that's one of the major problems. And the number of samples that we can record. Right now, as you saw, that we are for these papers, we have a number of samples about less than 10, right, for a group, because it's harder to do that type of experiments. But this limits the amount of information that we can get, because we need to have more human variability to make it to be more clinically relevant. So the last paper that we're putting together, actually, we are having 100 individuals, 50 with schizophrenia, with 50 controls that we get from 3 different brain banks. And this type of data is very, very valuable. So we are expecting to do more collaborative work with other groups in order to increase the number of samples that we can record and the level of analysis that we can do. Because we are right now just doing the minimum that and we know we can do more by kinetic analysis. Now, once we have this information, the next steps are logical steps is connect the synaptic dysfunction that we are measuring in people that donated their brain with synaptic dysfunction in people that are alive. And to do that, we need to do integrate electroencephalography with synaptic dysfunction. And we are starting to look for these collaborations. And some people are already starting to measure excitation to inhibition. Dr. Babiloni, that is going to be presenting, he will show how the electroencephalography information possess data about excitation and inhibition balance. So this is going to be very, very relevant to determine how the different brain regions are in balance in alive people, so early treatments can be put in place.
All right. Thank you very much. There is another question for you. Raluca says, hello, thank you for your presentation. My question is, how much can trauma affect the changes of receptors and the levels of excitability and inhibition?
Thank you. That's an excellent question. Yes, actually, a lot. Trauma is one of the risks for developing neurodegenerative diseases. And you know, there is a trauma associated in boxers or people that plays American football that is associated with dramatic behavioral alterations. And so trauma produced alterations in neuronal communication and in synaptic dysfunction. And actually, there is a work in Texas, in the Mitchell Center, where the repetitive trauma causes increase in toxic proteins like tau oligomers. Tau oligomers are actually the toxic aggregated proteins that seems to be the cause of tauopathies like Alzheimer's disease, frontotemporal dementia, and other tauopathies. And because they modify the synapse, then you can have those alterations. So yes, traumatic brain injury even sometimes mild traumatic injury or one strong, so mild repetitive or one very strong can produce alterations in the synapses. So that is another question that we have not answered. Other people have not answered yet, but this will be a good technique to try.
Right, thank you. And perhaps one final question. There is another one. So Raluca asks a follow-up question. As people heal from trauma, can their synapses change as well?
Yes, so this is an important question that, so we know that in schizophrenia and Alzheimer's disease, the changes are there. But we are, as I mentioned at the beginning, we are losing synapses all the time. So it's not that we are losing just the synapses, it's how the imbalance is there. And it's a chronic problem on these synapses that is producing the changes. So it's a functional alteration. However, if we can control the imbalance and the chronic challenges that the synapses are receiving, then we can give time or a window of time for synaptic plasticity to enter. In autism, we know, for example, that we have alterations of synapses, but behavioral treatments and behavioral interventions help to modify the signal-to-noise ratio that we observe in the synapses and actually modify the behavior, and the outcomes are very great. And this is because synaptic plasticity. So we are able to remove the imbalances and remove the chronic challenges, then plasticity will have time to enter and help to improve the behaviors in not only in trauma, but in other neurodegenerative diseases.
All right. And maybe really final question this time. Someone is asking about how targeting synapses directly would compare as a treatment strategy with targeting other factors such as microglia, for example.
Yes. So there is an interesting relationship between excitability and glia. So glia and excitability are intrinsically correlated. So the more excitability actually can lead gliosis. So people with epilepsy that have strong bouts of hyperexcitability have reactive gliosis. So, and in the other way, gliosis produced by bacteria or by toxic proteins can lead to alterations in excitability. How? For example, alterations in transporting in astrocytes that remove glutamate from the synapse can increase the level of glutamate to produce a hyperexcitability. And the opposite is true. So if you can have excitability, then the release of glutamate is going to be more. The enzymes are not going to be very good to remove all those things. And then you can have migration and activation of astrocytes and microglia. So it is important to determine what is the primary cause and treat that cause. So you allow the counterpart to work, because GABA and AMPA receptors are good targets. So for benzodiazepines, that classic target benzodiazepines and other medications work on AMPA and work on GABA and AMPA receptors. But it is important to remove the principal challenge.
All right. Again, thank you very much for the talk and for the interesting discussion.